second year MBBS class. Today we are going to start another new topic of biochemistry, and this is the introductory lecture for the topic of biochemical genetics. This topic has been divided into many subclasses. Like if you see the TOS, then you will see the distribution of MCQs and SCQs which you have to do in your examination, both the MCQ and the SCQ. And to get in the lecture, we are going to divide it into three phases. First phase includes A, B and C parts. Second phase included transcription and translation. And the third part included F, G, H, I and J. From the third phase, in the F, G, H, I and J, you have already learned the H portion in terms of oncogenesis, oncogenes. So today in the introductory lecture, we are going to see what are biochemical genetics and how we are going to learn different aspects of biochemical genetics in biochemistry. So in biochemistry, we are going to study basically the central dogma of molecular biology in detail. What is this central dogma of molecular biology? This central dogma of molecular biology is the conversion of DNA into RNA, which is known as the transcription, and then the conversion of RNA into proteins, which is also known as gene expression. Whenever gene is going to be expressed, it is expressed in the form of proteins. So this is the overall basis of biochemical genetics. When we go into the details, we encounter many different topics. Like in the first phase, we will see in the A part the structural basis of cellular information. What are the structural basis of cellular information? This structural basis of cellular information is basically two distinct types of chemicals or biomolecules, which are nucleic acids. One is known as the DNA and other is known as the RNA. And whenever these structures are going to be arranged in the cell, it is arranged in the form of DNA in case of eukaryotic systems. And RNA is also present in many other species. So the structural basis of cellular information is the DNA and RNA. In this subheading, we will see how this DNA, which is double-stranded, is arranged in the antiparallel strand and undergoes a process of replication, which is the doubling of DNA. And RNA, which is single-stranded, is going to be first converted into complementary DNA with special class of enzymes known as reverse transcriptases. And then we will become able to convert this DNA into RNA, and then we will complete the central dogma of molecular biology, which is the formation of proteins or the expression of the DNA in the form of proteins. So, in the first phase, we will see A, B, and C parts. A part will contain DNA and RNA, and the B part is the organization of this DNA. Whenever a large content of DNA has to arranged in the little space of the nucleus, it is going to be arranged or organized in the form of chromosomes. So these chromosomes are of different types, having different classes. We have two classes of these chromosomes, like autosomes and sex chromosomes. Autosomes are all the pairs of chromosomes except one pair, which is known as sex chromosome, and this sex chromosome is X and Y chromatids in case of males, while in case of females it is X and XX. To study different chromosomal abnormalities, we will see different types of techniques to study the chromosomal abnormalities. The study of chromosome is known as keryotyping. And in the C part, we will see how DNA is going to be replicated. The DNA is going to be replicated first in the semi-conservative mode of replication. As the DNA consists of two strands, one strand is conserved for the next strand synthesis. And this is how we will get two pairs of DNA from one pair of DNA after one round of DNA replication. There are many different types of reactions involved, and during these reactions we will have different types of components which are in the form of proteins, and all the processes and all the reactions and all the steps of replication is entirely different in case of eukaryotes and prokaryotes. So in case of eukaryotes, we will encounter different components, and in case of prokaryotes, we will encounter entirely different types of